Welcome to Biology Made Easy. Today we are discussing still diffusion and adaptation of respiratory and absorptive surfaces, because respiratory and absorptive surfaces do a lot of diffusion. So, what are examples of respiratory and absorptive surfaces? We have here respiratory surfaces, the alveoli, fish gills, toad skin, mesophyll spongy layers of the leaf we will discuss. Then absorptive surfaces, villi and microvilli of the ileum, nephrons, that's the kidney, root hairs, and placenta. Good. What is a respiratory surface? A respiratory surface is a surface where rapid exchange of gases, oxygen, carbon dioxide, okay, by diffusion between external environment and the cell. When the external environment comes in, then it's a respiratory surface. Then absorptive surface is a surface where rapid diffusion moves solids, water, mineral salts, dissolved food substances from the external environment into cells. Good. Now, the characteristics of both respiratory and absorptive surfaces is the same. Both surfaces are large surface area to volume ratio, very large surface areas, thin membranes, moist surfaces, highly vascularized with blood capillaries or channels that will carry away the diffused molecules or substances to other cells and tissues. So we'll look at some examples of respiratory surfaces. Alveoli. Well, the lungs, this lungs is made up of alveoli. This alveoli. Now, if we take out the trachea, bronchus, and bronchioles, what is left is alveoli that makes up the lung tissue. Now, these alveoli are adapted to be able to do gaseous exchange. So the alveoli have all these characteristics. Large surface area. The large surface area of the alveoli is brought about because there are numerous air sacs. They are not only numerous, but look at it, they are sac-shaped also to increase the surface area. So numerous, sac-shaped, increases the surface area. And the volume, they are thin. They are thin cells. They have thin cells. It's only made up of one layer of thin cell, that's an alveolus, one layer of thin cells, and the thin cells are epithelial cells, and so that gases can move through easily, and then the surface inside the alveolus is moist, so that gases can dissolve in and move into the cells. And then the alveoli are highly vascularized. They have blood capillaries that nerve it completely, surround the alveolus. And these capillaries take away the gases that have diffused into the cells. So that is it. It meets the characteristics of a respiratory surface. Gases in, gases out. Now let's look at another respiratory surface. That's the fish gills. The fish gills also do gaseous exchange. Oxygen in, carbon dioxide out. You see, the gill is made up of filaments. Let's look at this one. It has all these are filaments. But it has this bony structure, that's a bony arc that supports the filaments. And it has gill raches. These are the raches. They sieve out food. These are raches. Look, very short ones here. But the adaptation is that do the gaseous exchange is the gill filaments. These are them. They are numerous. And so they present a large surface area. They are thin. And so large surface area to volume ratio, they meet it. They have thin membranes that will allow gases to diffuse in. Well, the surfaces are already moist for the gases to dissolve in. And they are highly vascularized. Each filament has a lot of blood capillaries nerving it. and see the filaments. This is one gill with all these filaments, 70 filaments per gill, and each filament is highly vascularized. Each filament also have lamellae structures across it that also increases the surface area. So the gill filaments also meet the characteristics of respiratory surface very clean. We look at another respiratory surface and we look at the toad skin. This toad, you see, their skin is very loose on the body. It's loose, it's highly vascularized, has a lot of blood capillaries nerving the skin. This is outside the skin, but inside the skin is highly vascularized with blood capillaries. And the skin of a toad is always moist because gases dissolve in the moisture on the skin and then diffuse into the blood capillaries underneath. So it also meets the characteristics of a respiratory surface. That's toad skin. Now let's look at the leaf here and look at how it also meets the characteristics of a respiratory surface. Well, we have the stoma here. It's a hole so that the gases can move in, very neat. And then the gases can move in and out through this hole. But look at these spongy cells. These are the spongy mesophyll. They have large intercellular spaces. And so the cells plus the large intercellular spaces creates a large surface area to volume ratios, thin. And then also the cells are thin. And then also the spongy cells, the cell walls are moist so that gases can dissolve in and move into the cells and will move from cell to cell. And the spongy cells also have veinlets and veins, vessels that will also carry some of the dissolved gases in vessels to other parts of the plant. Now, we want to look at absorptive surfaces, which also have the same characteristics: large surface area, thin cells, moist surfaces, well supplied with blood capillaries. This is the digestive system. You have the small intestine here. That is where the digestive substances are absorbed. The ileum is the absorptive surface. Now, the ileum, the inside is these villi. The cell wall is lined with these villi, these finger-like structures. And all these villi increases the surface area for digested food substances to move in through all these spaces. So we have digested food substances here, and they are moving. All these areas are villi. And the villi doesn't only have villi, they have microvilli. So you have one villus like this, where food substances are going to move and move through. And not only the food substances moving from this lumen into the villi, right? But then the villi also have microvilli, where the food substances will move in through. And so large surface area, thin cells, the surface is already moist and is richly supplied with blood vessels, capillaries. Very neat. We want to look at another absorptive surface, which is the nephron of the kidney. Now, these are the two kidneys, one here, one here. When you look at the section through a kidney, this is a section through a kidney. You have the cortex, you have the medulla, you have the pelvis. This section, you see these structures which are called nephrons. These are tube-like structures nerved with blood capillaries. And each nephron is very long. And the whole kidney has about 1 million of these tubes, right? 1 million. And each of these nephrons is about 3 centimeters long. And all the nephrons together in this one kidney is about 120 kilometers long of nephron. And this nephron, you see how long it is? It presents a large surface area. Blood flows into each nephron, and the substances in the blood diffuse into this nephron. This nephron is a tube. And when the substances are diffused in, some of them will have to diffuse back into the blood capillaries to be absorbed back into the blood system. That is the shape of the nephrons, the number of them, numerous, large surface area. They are thin membranes. They are already moist. They are already well supplied with blood capillaries, absorptive surfaces, the nephron of the kidney. Then we look at the placenta. The placenta is a structure that is formed between the fetus and the uterus of mother. Now, so this is the placenta of the baby, is joined to the placenta. The umbilical cord is part of the placenta, we can say. But look, the placenta is a structure that is well separated and divided into small parts to present a large surface area to volume ratio. Here is villi, right? That is of the baby's blood system. All these are villi. All these are villi. All these are babies. All these are the fetus, for the fetus, blood capillaries, villi. And this is mother's section, also well separated, divided up. And this is a cavity that contains blood. Mother's blood and fetal blood does not mix to present a large surface area to volume ratio. And thin membranes so that substances can easily diffuse across, and very well supplied with blood capillaries, absorptive surfaces. They all meet the characteristics. Now we look at the last one for today. We look at the root hairs. The root hairs are also excellent adaptation to absorb mineral salts and water. Look at this root. It has all these finger-like structures, are hairs. They are root hairs. This root is going to absorb substances like phosphates and other substances like magnesium, salts, and they will have to absorb it all over. Water too will need to go in. So the root hairs present a large surface area so that each of these small pieces is absorbing something. So we have here, this is the outermost layer of the roots, the pyliferous layer, if you like, the epidermal cell. Each epidermal cell produces this extension, that is the root hair. So this surface, all these surface absorbing substances increases the surface area for absorption. And the cells is so thin for substances to move in, and outside all here is moist. So the substances that are moving in, the magnesium salts, the sulfates, they are all dissolved in the water to move in here and large surface area. And these cells are thin. Other epidermal cells will be thin to move the cells from cell, the substances from cell to cell, until it gets to the vascular tissues to move the substances up. Very neat. So this brings us to the end of our looking through the characteristics of absorptive and respiratory surfaces that diffusion plays all the parts to move substances. So till we meet in our next lesson, thank you very much and goodbye.